You know, when scientists get a sample of something complex, like, say, river water or a plant extract, how do they figure out exactly what's inside? Well, they use this really elegant technique called chromatography. And honestly, the best way to think about it is as a kind of race, the great separation race. So here's our game plan. First up, we're going to break down what this race is all about. Then we'll look at the original method using just simple paper. After that, we'll see how scientists scale things up with columns, and then check out a faster, more precise, modern version. And finally, we'll connect all of this to how it's being used in our world every single day. All right, let's dive into section 1, the Great Separation Race. Okay, so here's the big question, right? You've got a mixture and you need to see all the individual parts. Think about a single dot of black ink. You kind of know it's made of different colors, but how do you actually pull them apart and prove it? That is exactly the problem chromatography was invented to solve. It's all about unscrambling these chemical mixtures. Now, the official definition can sound a little technical, but the core idea is actually super simple. Just stick with that race analogy. You have a bunch of different runners, those are your chemical components, and they all start at the same line. When the race kicks off, some are just naturally faster than others, so they start to spread out. Chromatography is really just the science of organizing that race. So, in any race, you need two key players. First, you've got the stationary phase. Think of this as the racetrack itself. It literally stays put. It could be a strip of paper, or maybe a fine powder like silica. Now, some of our chemical runners are really attracted to this track. They want to hang on to it. Then you have the mobile phase. This is the thing that gets everybody moving. It's like a strong wind at the runners' backs. It's a solvent that flows over the racetrack, pushing all the components forward. The magic happens because some runners get pushed along really easily, while others get slowed down because they keep sticking to the racetrack. All right, so where did this whole idea even come from? Well, let's jump back to 1865 and talk about the beautifully simple technique that kicked off this entire field, paper chromatography. The process here is just so intuitive. You start by putting a tiny spot of your mixture onto some filter paper. That's your starting line. Then you dip the edge of the paper into a solvent, our mobile phase. The solvent starts to creep up the paper through something called capillary action, just like how a paper towel soaks up a spill. As soon as that solvent hits the starting line, the race is on. It picks up the mixture and carries it up the paper. The components that don't really care for the paper and love the solvent, they travel way up high. The ones that stick to the paper lag behind. And hey, if your runners are invisible, no problem. You can use things like a UV light or a chemical spray to see where everybody finished. So how do we score this race? Well, we use something called the retardation factor, or just the RF value. It's a really simple ratio. You just measure how far one of your components traveled, and you divide that by how far the solvent traveled in total. This gives you a unique score. And here's the cool part. As long as the conditions are exactly the same, a specific chemical will always have the same RF value. It's a fantastic little fingerprint for identifying substances. Okay, paper chromatography is great for analyzing a tiny spot. But what if you need to separate more than that? I mean, what if you actually want to purify a larger amount of something? For that, we've got to scale up. So let's talk about column chromatography. With this method, our racetrack is inside a glass tube, or a column, and you have to carefully pack it with a material like silica. Now, getting this right is absolutely crucial. You need a perfectly smooth, uniform track. Any air bubbles or cracks could totally mess up the race and ruin your results. Once the racetrack is all set, you carefully load your sample right on top. Then you add the mobile phase, and gravity does the rest, pulling everything down through the column. And just like that, the race begins. You can actually watch the components separate out into these distinct bands of color moving down the column. The fastest runners, the ones that don't stick to the silica much, they reach the finish line at the bottom first. And by collecting the liquid in different test tubes over time, you can actually capture each component one by one as it finishes the race. Okay, so we've seen the original method on paper, and we've seen the scaled-up version in a column. But what if you want the simplicity of paper, but with way more speed and precision? Well, that's where the modern evolution comes in. Thin layer chromatography, or TLC. Now, TLC works on the exact same principle as paper chromatography, but think of it as a serious upgrade. Instead of a paper track, you've got this perfectly thin, super uniform layer of silica coated onto a glass plate. This makes for a much more consistent racetrack, which means your results are sharper, cleaner, and way more reliable. The process is the same, spot the sample, add the solvent, but the separation happens faster and better. It's the perfect tool for a scientist who needs a quick answer, like, hey, is my reaction done yet? Or just how pure is this sample? So we've covered the how, but now it's time for the most important part, the why. Why does running these tiny little chemical races matter so much in the real world? So let's do a quick recap of the methods. You've got paper chromatography. It's beautiful, simple, perfect for a basic ID. Then there's column chromatography, which is the real workhorse you bring out when you need to separate and purify a whole bunch of something. And finally, thin layer chromatography, which is all about speed and sensitivity, perfect for those quick purity checks. You see, each one is a different tool for a very specific job. And you would be amazed at where these separation races are happening. I mean, they're everywhere. In the pharmaceutical industry, chromatography makes sure the medicine you take is pure and safe. In food science, it's used to check the vitamins in your cereal. In forensics, it can identify a mystery substance from a crime scene. It really is an invisible pillar of modern science, working behind the scenes to keep us safe and push our knowledge forward. So really, the whole thing boils down to this. It's all about controlling the race. By carefully choosing our stationary phase, the racetrack, and our mobile phase, that push that gets everything moving, we can basically custom design a race that will separate pretty much any mixture you can think of with incredible precision. And this ability to deconstruct our world into its most basic parts it's incredibly powerful. It lets us understand things, purify them, and create new things. Which really leaves us with one final thought. Now that we've mastered the art of this great separation race, what new frontiers is it going to let us explore? What discoveries are just out there, waiting to be separated?